Hello everyone. Today we're going to talk about microbial growth conditions, and we're specifically going to talk about temperature. How can the temperature affect the bacteria growth? So let's go ahead and get started. So let's go ahead and take a look at the bacteria and how we categorize them based on their temperature requirements. So you see right here, there's a graph. We're graphing basically the temperature down here on the x-axis, and on the y-axis, we're graphing the growth rate of the bacteria. We have several terms: psychrophiles, mesophiles, thermophiles, and hyperthermophiles. So we'll talk about each one of them. We're also going to discuss the optimal temperature. So as you can see, for each group of bacteria, basically those are the terms, by the way, that we give to the bacteria. So we have bacteria that are considered psychrophiles, we have bacteria that are considered mesophiles, etc. And for each one, there's a range. Okay, so for example, for mesophiles, you can see that these bacteria, they can survive, negative, sorry, not negative. They can survive at 15 degrees Celsius all the way to around like 45 degrees Celsius. So it's a range, okay? But there's the optimum growth. And so right here at the very peak, okay, this would be considered the optimum temperature. So mesophiles, they would do the best, okay, at around, let's say, 35 degrees Celsius. Okay, psychrophiles would do the best at around 10 degrees Celsius. Thermophiles, they do very well around 65 degrees Celsius. Hyperthermophiles, around 95 degrees Celsius. So that's the optimum temperature. So basically the optimum temperature would be at the peak, right? At the very high point, okay, of those peaks. But as I said, for each group, there's a whole range. So let's talk about mesophiles first. So mesophiles, those adapt to moderate temperatures, because you can see they can go from 15 to around 45 degrees Celsius. And humans, right, our body temperature is around 37 degrees Celsius. So bacteria that can live inside of us, they are mesophiles, okay, because 37, as I said, is our, body temperature. And as you can see, this is kind of like, again, around the optimum, right, temperature for mesophiles, and that would be our body temperature. So things like E. coli, Salmonella, Lactobacillus, those are examples of mesophiles bacteria. Now, psychrophiles, they prefer lower temperatures, right? So they are more on the colder, okay, temperatures. And so you can see right here, they can go like negatives, right? So they can even tolerate negative temperatures to around 20 degrees Celsius, okay? So, and by the way, like the fridge, right? The fridge temperature is around like 4 degrees Celsius or so. And so therefore, these psychrophiles, those are the ones responsible for food spoilage. Okay, so even though you put your food in the fridge and then unfortunately it still goes bad, well, you can blame the psychrophiles for that. Okay. Now we also have thermophiles. You can see that thermophiles, those are the ones that actually prefer hotter temperatures. Okay, so you can see that thermophiles, they prefer 42 degrees to around like 80 degrees Celsius, and that's pretty hot. So those are definitely heat lovers. And so you would find them in hot springs, okay, for example. And so they even use them, by the way. They use thermophile bacteria to help with garden composite. So to help basically break down kitchen scraps, okay? they add thermophiles to help break down, kitchen scraps, and they kind of create it in a way to make it, again, like, high temperatures or whatever, so it works. And so hyperthermophiles, let's talk about those. Those are, they love insane heat. So, 65 degrees to around 100, 110, 105 here, 110. Some of them even go like even higher, okay? So hyperthermophiles, they love extreme high temperatures. And so those, you would find them at hydrothermal vents, for example, at the bottom of the ocean. And so they would be able to survive such high temperatures. Okay. And so those are basically some examples, okay, of the different groups of bacteria. By the way, when we try to kill bacteria in the lab, we usually take our old plates and we stick them in what we refer to as an autoclave machine. And this autoclave machine can actually, temperatures in an autoclave machine can go beyond 121 degrees Celsius. And so the reason why we try to make the temperature like super high so that we can try to kill, a lot of the bacteria. Now, as I said, some hyperthermophiles, they can even survive that temperature though, but, that's kind of what we use to try to get rid of and kill all the bacteria in our lab. We use the autoclave machine. Now for the virtual lab, what you're going to do is you're going to take a bacteria, okay? So this is a broth right here, and there's bacteria, cultured in that broth medium, and you're basically going to go ahead and you're going to place, you're going to like inoculate, some of that bacteria into like your broth medium, and then you're going to go ahead and stick it at 5 degrees, 55, 42, 38, and 25. You're basically going to see what's going to happen, right? Like you're going to see how well is your bacteria going to grow at 5 compared to 42, compared to, room temperature. And based on that, you're going to be able to tell whether your bacteria is a thermophile, hyperthermophile, whether it's psychrophile, etc., mesophile. And so you're going to basically determine, as I said, the different temperatures, okay, in which the bacteria can grow. And then how, after you go ahead and you actually pick up your media out of, the incubator. So here, this is like the 5, this is the 25, 38, 42, and 55. Notice how the turbidity is different. Okay, so notice how right here the broth is more turbid. Remember how we said turbidity is an indication of bacteria growth. So the more turbid your broth, the more bacteria growth you have. And again, that's going to help you determine whether it's a mesophile, whether it's a psychrophile, etc. And so one way actually to go ahead and read the turbidity, okay, other than just kind of looking at it and realizing just by comparing those two tubes, this one is way more turbid than that one, for us to get accurate measurements, we can use something called the spectrophotometer. And the spectrophotometer, this is an image right here of a spectrophotometer. It's basically a machine where you take some of your sample, but this whole entire tube is not going to be able to fit because you open this compartment and this tube is not going to fit in there. So you take a sample and you put it into this little, tube right here. We refer to it as the cuvette. Okay, so this is a cuvette. You take the cuvette filled with the sample and you stick it in there, and then you close up this compartment. And what this machine can tell you, well, it can tell you two things. It can tell you how much light is absorbed and how much light is reflected off. So what happens is when you place the cuvette in there, there's a light source. Okay, so the light basically hits the cuvette. Okay, and when the light hits your cuvette, which basically has the sample, right? Some of the light is going to be absorbed. So it's going to give you that number. Okay, and then some of the light is going to be actually reflected off. And it's also going to give you that number. Okay, so it gives you two numbers. How much light is absorbed, how much light is reflected. And actually, the more concentrated your solution is, okay, the more light is going to be absorbed, and therefore the less light is going to be reflected off. And that's how you can tell, you compare the numbers, okay, for each tube, and based on the numbers, you can tell which one is more turbid and therefore has more bacteria growth based on how much light was absorbed and how much light was reflected off. And so the numbers that you get, as I said, those like absorbance numbers and reflective numbers, we call it optical density measurements. And as I said, it's basically going to give you information, how much light is absorbed, how much light is reflected off. And again, this can help you determine, okay, which tube has the most bacteria growth, and therefore you can tell which temperature range would be preferred by this bacteria, okay? And as I said, this one is called the cuvette. Okay, and the machine one more time, the spectrophotometer. You might have actually used it, by the way, in general bio classes. Okay, we usually use it. And as I said, we take a sample, okay? You don't stick the whole entire tube in the spectrophotometer. You take a sample. How are you going to take a sample? Are you going to pour it in? No, you don't want to spill bacteria all over. So we usually use our pipettes, okay? And you can take like 1 ml or so of the sample, place it in the cuvette, place it in the spectrophotometer, get your measurements, okay, of the turbidity of each tube, and based on that, you will figure out the temperature range. And for the worksheet, you're going to take a look at these results right here. Again, you're taking a look at a bunch of, broth media, and they're inoculated with E. coli. And again, when you see turbidity, that would mean there's lots of bacteria growth. So take a look at the temperature ranges, and based on that, figure out, again, whether they're mesophile, whether they're thermophile, hyperthermophile, psychrophile, etc. Okay, and that's another set of results, same concept. Okay, you're going to take a look at the temperatures, and based on the results that you see, figure out what is the range, okay, and therefore what would that bacteria be called, okay, based on the temperature range that it prefers.